Well, it was in my childhood days. Oh, Lord, a minute.
Welcome to The Balancing Act. I'm Michelle J. Buckle, your host. I'm an audio lover, climate campaigner, an artist, a storyteller, a healer, and your friend. Today I will look behind the scenes to learn more about transgenerational plasticity, which occurs when offspring is influenced by its parent's environment. We'll also look at transgenerational plasticity's relationship to stress and the implications of their findings across our ecology. In my explorations, I've learned that major depression, anxiety, and post-traumatic stress disorder, among other stress-related psychiatric disorders, are highly prevalent in certain populations. And these stress-related illnesses sometimes get passed down from one generation to the next, and then the next. Well, this discovery led me to wonder, can the long-lasting stress of parents affecting their offspring also impact subsequent generations? If so, then these stress-related disorders may become in our future the leading cause of disabilities across the world, which in turn will demand increasing medical, economic, and environmental resources. In a 2021 study in Molecular Cell, scientists Dr. Das, Dr. Min, and Dr. Prahlad from University of Iowa studied roundworms and how maternal stress can have long-lasting epigenetic effects on offspring. With the discovery of the ability of genes to alter their expression in response to the environment, modern science recognized what artists and psychologists have known experientially. The body remembers. Hello, Dr. Das. Welcome to the show. We are so grateful to have you here with us. I wondered if you could share with our friends at the Balancing Act what led you to study roundworms and maternal stress.
The topic of our research is how the memory of stress is transmitted to the one from one generation to another. Now, the best model would be human, because ultimately we want to know what happens in human. And but the thing is, as you know, it's hard to do this in human, because I can't purposefully stress a pregnant mom and see what happens to the children. That I can't do. And the other thing is that human live for like 70, 80, 90 years, and like maybe I'd say in 30, 40 years I'll see the children, and I want to wait till the children get adults and what happens to the grandchildren. So that's technically not possible to do this exact same study in human. So that's why we chose roundworm, which is very handy and very user friendly model, because they live only for 20 to 30 days, and in 4 to 5 days you get their progeny, meaning you get they lay the eggs and the eggs mature. So in 4 to 5 days you get their progeny, and you can, so studying this is very easy and practical to do this in the roundworm. Now the question obviously is, whatever we are seeing in roundworm, should we also see the exact same thing in human? The answer is, I don't know. Honestly, I don't know at this point. So some of the features that we see in roundworm are seen also in human, but not all. So for example, like the roundworms, they have nervous system, they have neurons, they have intestine, they have germline, which is very much similar to human. But then, of course, in the course of evolution, human evolved and the roundworm didn't. So there are certain differences at the DNA level. There are big differences between roundworm and human. I mean, that's why we are human and those are roundworm. So I'm not 100% sure that what we are seeing in roundworm will see the exact same thing in human. But because the proteins that played a crucial role in our study are conserved in human, so the same protein exists in human. The protein is very much conserved. It's evolutionary conserved, and its function is conserved in across all the organisms like Drosophila and like xenopods, like frogs and Drosophila and yeast and human. So this protein is very conserved. So that's why we are hopeful this function what we see in the roundworm. We are hopeful that it's the same thing in human. as you already mentioned, that there are reports in human that the mothers who are stressed when they were pregnant or like after they conceived, then their progeny, like their child, have some developmental defects, sometimes like metabolic disorder, neuropsychiatric disorders like schizophrenia or autism. So there are reports like that which sounds very similar and interesting, and like that's our hypothesis that this is the reason. I mean, this is the same thing is also happening in human, but just doing the research on human is not possible. So that's why
Yeah.
we.
Okay, that makes so much more sense. I'm interested too, though, because not only are you seeing that there is a shift in terms of, I think you said something along the lines of the response. I'm interested in how the epigenetic changes are triggered by stress, build resilience that's passed on. So not only when you talked about there is a negative effect that happens, you also talked about this positive thing, and that means that they're preparing the offspring so that if there's a risk factor that happens, they might be able to overcome that.
Yeah. So what we are seeing is the progeny, they are more stress resistant. So for example, I mean, in our case, we used heat temperature as a stress. So these roundworms, they usually grow at 20 degrees, their normal temperature. Now we expose them to 34 degrees centigrade. So at a very high temperature. So that's a stress, like a thermal stress for them. So what we see is when we expose the mothers to higher temperature, then their progeny are more stress resistant, meaning, so
Yes.
if we keep the worms at higher temperature for prolonged time, then they die. But the progeny, they don't die or die much comparatively like less to to high temperature if they come from a stressed mother. So they're more stress resistant, what we call resilience. So they have this sort of a gain of uh, function, meaning they're more stress resistant. So that's the plus side, like that's the advantage. Now the disadvantage is they are in roundworms, sometimes these progeny, they are sterile, they don't lay eggs, and sometimes they have some developmental defects we see in the progeny. And the other interesting aspect of our story, I think, is depending on the dose or intensity of maternal stress, this inheritance of resistance is transmitted to, like, so if we transmitted across generations, meaning if we hit shock the mother only for say 5 minutes, like for a
Yeah.
shorter time,
Yeah.
then only their immediate progeny, only their children are stress resistant, but their grandchildren are not. However, if we heat shock the mother for like a 60 minute, like a 1 hour heat shock, then we see that even their grandchildren are also resistant.
Ah, yeah.
So
That's
that
like
is,
complex trauma. It's
yeah.
like
So
layered.
right. So the higher the dose or the duration or the intensity of stress, the longer it exists over the generation.
I just had to really breathe in what you just said. Wow.
Like,
This is
isn't it like fascinating? I mean,
it's fascinating.
when we first saw this, we are very excited to see this, that not only it's transmitted from like mother to progeny, but depending on the intensity or duration of stress, it that dictates how long it will exist, meaning across one generation or the two generation or so. So that was important. And the thing is, I think why our study is important, or why is it exceptional from many similar studies, is that we have some mechanistic understanding, insight, like at the molecular level, what happens, what dictates this inheritance of stress resistance, that we, to some extent, we could uncover that mechanism.
Yes.
So this
Yes.
epigenetic, meaning like something that changes the DNA. So because DNA is the genetic material, DNA is what is passed from one generation to other, like whole the genetics, Mendelian genetics and everything says DNA is the genetic material, meaning from parents to offspring, DNA is transmitted. But there are some differences, like DNA is not changed, but I would say DNA is modified, you could say, and that modification is transmitted to the DNA. So we found one such modification in a very specific location, which is responsible for this stress resistance. Does that
This is,
make sense?
yes, it does make sense. So I'm going to tell you a personal story because it connects. So I told you that I started this journey because of my nephew being murdered. I'm interested in the transgenerational impact of homicide and loss. My parents grew up in Jamaica and were brought over on, originally slave ships in terms of our history from Africa came on slave ships. So they were exposed generally in Jamaica to slavery, right? And I had such a strong impact to my nephew's loss. And so as I did my own personal work, I started to ask, what was that all about? And I started to realize that I have been impacted by the fact that I can't have children. Yeah. So I have been sterile. So I've had this query about the transgenerational impact of trauma that might have happened earlier that's come down to me now in this situation where I can't have children. So it's no coincidence that I found you in this study and to hear that there's this particular area within the roundworm that you said actually is being affected.
So that is very, like, I don't know, I don't have the right word. I mean, in terms of science, it's very fascinating, but maybe fascinating is not the right word. That you didn't experience the stress by yourself, but somehow so that is what we call the memory of stress. So
Yes.
there is the memory of stress that is transmitted to next generation who are not exposed to stress. Their parent was exposed to stress, but somehow they carry the memory of stress,
That's
and
right.
that alters their physiology, that alters their behavior.
Okay. So if we're going to take this learning now and we're going to understand it for the larger population, what would you say is the message that you're hoping that you would extract from your research that would support our world and society or make sense of or get us to ask questions about?
So I think the next, I think what we, our study only like set up the building stone, or like we only did we are only at the first step, because obviously in the in a human system, the same thing, I mean, we might see just by observation, we see similar things, but it must be at the molecular, at the cellular level, at the molecular level, it has to be much more complex. But at least from our study, we have some hints where to start from. If we don't have any hint where should we look at, then in human we have more than 30,000 genes, we have thousands of proteins, and then so it's very complex. I mean, in roundworm they don't have as many organs as we have. I mean, they don't have, for example, like they don't have heart, they do have nervous system, they do have intestine and muscle, but they don't have all other, like the organ system in human is much more complex. So I can imagine that the same thing, although we see the same similar thing in human, at the molecular level it's much more complex, so it's very hard to start without any hint, without any idea what should we look. But now, since we know, we have some information that this protein and like this particular protein A and protein B interacts and do this, so we can look at the human level, in the human. So we also do like human tissue culture cells in lab, I mean, we do that. Or we have like there are like mouse models where we can study this. So even if we can't do this in actual like human living
Humans.
humans, we can do this in human tissues, human cells, in laboratories, mice tissues. Sometimes people use post-mortem brain samples to look at different changes. So we can look if the same protein-protein interaction, what we see in the roundworm, exists in human. We are hopeful that it exists, but we are not sure because we haven't tested that. So that should be the immediate first step to see if this phenomenon is conserved in human.
So, and that's quite the job to do that. But
And the
you're
other
just.
thing is this
Mhm.
this particular protein, what is heat shock factor 1, is also very strongly implicated in cancer, in aging, and in many other processes. And so this protein interaction, meaning HSF1, the protein HSF1, heat shock factor 1, it interacts with another protein in roundworm, what we call MET2. This is a novel interaction. This was not known before, that HSF1 interacts with MET2. But and in human there is a there's a equivalent of MET2, equivalent enzyme. It's a different name, but it does the same thing. So one should look at in human if HSF1 and that protein also interacts and does similar things.
Okay, okay. I'm noticing that, especially in the Black community, there's a disease that's becoming a lot more common, a disorder called amyloidosis. Are you familiar with that?
Yeah,
And it's about a protein that's building up and being deposited in different parts of the body,
brain.
in the brain or in the kidneys or in the. Is this, could this potentially have some connection?
So the thing is, this age related, yeah. So these age related disorders, for example, Parkinson's or Alzheimer's or Huntington's, so they are considered as genetic disease because there is a genetic background. Like the people have the scientists have identified the genes which is responsible, but there are also very strong environmental effects that we don't understand very well yet, unfortunately, meaning the scientific community. Like, but this is what you just said is not, I mean, it's real, meaning certain populations, certain ethnic groups are either more susceptible or more resistant to certain kind of diseases. So I know like a certain population in Africa, they are very resistant to malaria.
Mm-hmm.
So that was very like that was amazing. I mean, their
Mm-hmm.
neighboring communities, they have the malaria, but that particular community, they don't have malaria ever. They people,
Mm-hmm.
scientists were interested, and they found that they found a difference in their DNA, what makes them resistant to malaria. So this is the gene and environment interaction is
Yes.
amazing.
Very amazing and very significant because there's a strong link between some of these diseases and stress
Yeah.
and cancer and stress. So toxic stress, which is my area of interest, was looking at adverse childhood experiences and the impact that it
Hmm.
has on the body. And how do we begin to learn about work that you're doing so that we can begin to shift things that are happening within us, be aware to make changes, healthy choices, or even, just understanding the impact of certain chemicals in the environment, like environmentally. There's so many different layers that are impacting us as an ecological system. And I think this is a wake up call. And this is what I'm
Hmm.
hearing from you.
Yeah, because this stress, like the environmental stress or any kind of stress, now at least we know, and we that it doesn't only impact that particular person, it impacts the next generations,
That's right.
which is very warming.
Yes, and very important for us because we're leaving them behind, aren't we? And we want to ensure that they have a safe and healthy place to be in.
Hmm.
Wow, this is awesome. I'm so glad this we are just having this amazing chat right now, and I just love the way it's going. And I just want to ask you one last question, and that is, I'd never found out about who you are other than to read about you online and where you came from and your background culturally as well as a human being. I just feel so blessed today as a diverse individual. I am proud. I am so proud of the work you're doing. So I'd love to hear a little bit about you.
Thank you very much. So I'm in here in University of Iowa. I'm right now working as a postdoctoral research scholar. So before that, so I'm from India, by the way. So I'm from India. I'm from the eastern part of India. So that
Mm,
state is called West Bengal. So that's in the eastern part of India. And my name is Srijit Das, you know. I did my like undergrad and masters and PhD, everything from India, and then I decided to come to US for further research.
So it sounds like what's next for you in the future would be what? What's your next steps?
Obviously, the next step should be to check what we see in the roundworm is relevant in human. That's very, very, very important, because if this mechanism is not conserved, this is not really very useful, because ultimately our goal is to improve human life, not necessarily like a roundworm life. I mean, yeah, I mean,
Have you got funding to be able to take that next step?
yeah, we are doing this. I mean, in our laboratory, we do have tissue culture like human cells on petri dishes. I'm testing whether the same thing happen in human, and if it does, then we will go to the next level of model organism, like maybe mouse and maybe postmortem brains of people who had experienced stress,
Okay, wonderful.
because that
Wow.
because really that's the most closest sample, right? If I can get samples from a human being, that will tell us the best information.
Well, you know what? This is awesome. It's fabulous. Um, just to have you have this conversation with me, Dr. Das. Wow, I am excited that I will get a chance to share this story online. And I honestly believe that this is going to make a big impact for us generationally. One of
Hmm.
the biggest things that I believe is that, the idea of, whether it be conscious or unconscious traumas that are unresolved, they resurface in the family system. And they can resurface in many different ways. And we're seeing it even in roundworms.
Hmm.
So, anyways, I just want to say thank you so much. So
Thank you
much for
very much.
today.
Thank you very much, Nichelle. It was
Yeah.
very nice talking to you and learn from you, knowing what like other people think about the scientific community. I know people have hope that we'll do something relevant for the greater people, the greater society, and we'll definitely we always try to do that. That's always our mission. That we hope that our research will improve the human health and society at someday, if not today.
Thank you, Dr. Das, for sharing the fascinating findings of your research on roundworms and how maternal stress can have long lasting epigenetic effects on offspring. Perhaps your type of research will be pivotal for understanding how not only unresolved trauma, but also resilient experiences imprint themselves in all our lives across our ecology. Oh, that is why it's so important that our communities continue to support and understand research into the risk and resilience of stress-related disorders, so that we might develop effective and preventative strategies for healing and transforming our ecology across the lifespan. Thank you. I'm Michelle J. Buckle, your host from The Balancing Act.